Hello, my name is Antoinette Adams, and I am a grad student here at Grand Canyon University. I am currently in class Bio 515, and today I will be presenting about the pancreas, insulin, and glucose. So, as an introduction, the pancreas is a digestive function. It secretes two important hormones. You have insulin, and then you have glucagon. They are both critical for normal regulation of glucose, lipids, and protein metabolism. Other hormones such as amenolin, the somatostatin, and the pancreatic polypeptide. So, what did I just explain? I just told you that the pancreas produces two essential hormones, glucagon and insulin. They both play a critical role in controlling blood sugar levels. By promoting the uptake of glucose into cells for energy or storage as glycagon, insulin, which is released by the beta cells in the pancreas, it lowers the blood glucose levels. In contrast, the pancreatic alpha cells secrete glucagon, which raises the blood sugar levels by inducing the liver to convert glycogen that has been stored into glucose and released into the bloodstream. So insulin and glucagon, they work in tandem to preserve glucose homeostasis, which guarantees a steady flow of energy for the body. So diabetes and other disorders can result from an imbalance in these hormones. So here's a diagram of the pancreas. The human body needs strict regulation of blood glucose levels to maintain normal bodily functions. The brain, the pancreas, the liver, the gut, as well as muscular tissue, it emits a complex network of hormones and the neuropeptides that work together to do this. The pancreas plays a critical role in this network by secreting the hormones insulin, which lowers blood sugar, and glucagon, which opposes it. The pancreatic hormones insulin and glucagon, they regulate metabolism. A feedback loop between glucagon and insulin maintains blood sugar levels within a specific range. When the level of glucose is too high, insulin lowers it. When the level is too low, glucagon raises it. Metabolic illness such as diabetes, which is an insufficient insulin production or response, or an excessive insulin or lack of glucagon response, they both can result from disruptions in the secretion or activity of these hormones. Additional pancreatic hormones include the pancreatic pelopeptide, which controls the pancreatic enzyme secretion and the gastrointestinal function, and the somatostatin, which induces both glucagon and insulin. So I received that information from Router 2016, which produced the data that we see here on the screen. If we look close to my clicker here, here's a photo of the human body. This is the pancreas. So you have this input and this output channel, which is where the glucose is raised and lowered. Next, we'll discuss the isolates of the Langerhans. So the pancreas is home to unique cell clusters called isolates of Langerhans. So those are simply just placed in charge of generating a number of hormones necessary for controlling blood sugar levels and the digestive function. Now we'll discuss the production of pancreatic hormones by three cell types. So you have alpha cells, they produce the glucagon. You have beta cells, they produce the hormones insulin and amylin. You have delta cells, which produce the hormones of gastrin and the somatostatin. You have F cells, they produce hormone pancreatic peptide. The hormone glucagon, which is mostly produced by alpha cells, are located on the outer portions of the isolates, which is essential for raising blood sugar levels when they fall too low. In order for glucose to be released into the bloodstream, glycogen, which is stored in the form of glucose, must first be broken down by the liver in response to a signal from glucagon. In order to ensure that the body has a consistent source of energy during fasting and between meals or during physical activity, this process is called glycolysis. It is critical and very important. So glucagon promotes the glycogenesis, the process by which the liver produces glucose from non-carbohydrate sources such as amino acids. Glucagon functions as insulin counterbalance, preventing the hypoglycemia, or which is simply low blood sugar. Two major hormones produced by the most numerous cells in the isolates of Langerhans are insulin and amylin. These are produced by beta cells. So beta cells are just probably the most well-known hormone that the pancreas produces. Insulin is essential for reducing the blood sugar level after meals. It makes it possible for blood glucose to enter cells where it can be stored for later use as glycogen in the muscles and liver. It is also consumed as fuel. Next, we'll discuss the hypoglycemia results, which is just an acclimation of glucose in the bloodstream caused by insufficient insulin production or an improper cellular response to insulin, in which this case would lead to diabetes. In addition to secreting insulin, beta cells also release amylin, which displays the emptying of the stomach and therefore lowers the pace at which glucose enters the bloodstream. Gastrin and somatotropin are two regulating hormones that are produced by delta cells. Gastrin increases the stomach production and gastric acid, which helps break down food and digestion easier overall, especially for proteins. Gastrin has a role in digestion and is mainly produced in the stomach, but the pancreas also contributes to its creation. So on the other hand, so simply what I'm saying is on the other hand, you have somatotropin, and that is an important hormone that inhibits the blood flow. In the isolates of Langerhans within the pancreas, there are three main types of hormone secreting cells. You have A cells that secrete glucagon, B cells that secrete insulin, and D cells that secrete somatostatin. The A and D cells are typically located around the perimeter of the isolates, while the B cells responsible for insulin production are found in the interior. As insulin is released from the B cells, it flows through the venous return and passes by the A cells, allowing it to directly influence them by inhibiting glucagon secretion. This close anatomical relationship ensures that insulin effects can modulate glucagon release, maintaining blood sugar balance more effectively. The pancreatic hormones, insulin and glucagon, regulate metabolism. So the insulin and glucagon balance in the body during two distinct metabolic states. The fed state, you have the left panel, and then the fast state is on the right panel. It's depicted in the figure below. So on the left are the insulin dominants. After eating, blood glucose levels rise and insulin becomes the dominant hormone. Insulin promotes several processes. Glucose oxidation is the conversion of glucose into energy. You have glycogen synthesis, which stores excess glucose as glycogen and delivered in the muscles. You then have the fat synthesis, uh, which stores excessive energy as fat in tissues. You have the protein synthesis, which builds proteins from amino acids for growth and repair. As insulin activity increases, glucagon activity is reduced, which helps store energy and prevent the release of glucose from the liver. So on the right, you have the glucagon dominance. During fasting periods or periods between meals, your blood glucose levels decrease and glucagon becomes the dominant hormone. Glucagon stimulates processes that increase blood glucose, the glycogenesis, breaking down stored glycogen in the liver to release glucose. The glucogenesis creates new glucose from non-carbohydrate sources like amino acids. You have the ketogenesis, which produces the ketones from fat breakdown as an alternative energy source, especially when glucose levels are very low. In this state, insulin levels are lower, which reduces glucose uptake and storage, allows glycogen to raise blood sugar and provide energy during fasting. We are now at the structure of insulin. Insulin is a polypeptide hormone composed of two chains, A and B. Both chains are derived from proinsulin, which is a prohormone. The two chains are joined by disulfate. I'm sorry. Specific targets of insulin action in carbohydrates. The increased activity of glucose transporters, it moves the glucose into cells. The activation of glycogen syntheses, it converts the glucose to glucagon. And then you have the inhibition of carbokinase, which inhibits the glucogenesis. The specific targets of insulin action occurs inside of lipids. The activation of oxaloboxylase, it stimulates the production of fatty free acids. The activation of the lipoprotein, it breaks down the fatty acids and then are then taken up and it's made and stored in the cell. So insulin facilitates the uptake of glucose and amino acids primarily in liver, in the skeletal muscle, and found in tissues. It acts on insulin receptors to trigger a cascade of cellular pathways, which enhances the location of glucose transporters to the cell membrane. In the absence of insulin, most tissues show a significant reduced capacity to absorb glucose and amino acids, which then leads to elevated blood glucose levels and impaired nutrient utilization. So, insulin promotes glycogenesis, the process by which glucose is converted into glycogen for storage in the liver and muscle. This process is essential for maintaining energy balance, especially during fasting or between meals. Next, you have the lipid synthesis. In tissue, insulin stimulates the synthesis of lipids from free fatty acids. And so this action pretty much just helps store the excess energy and is essential for maintaining healthy body fat stored in the body. Regulation of insulin release. So the major stimulus, you have increased blood glucose levels. After a meal, your blood glucose will increase. In response to increased glucose, insulin is released. Insulin causes uptake of glucose into tissues, so blood glucose levels decrease. Insulin levels decline as blood glucose declines. Targets of the glucan action, it activates, which cleaves off a glucose of phosphate molecule off of the glycogen. It inactivates the glycogen synthesis by less glycogen synthesis. It increases the carbokinesis, which stimulates the glycogenesis. It activates the lipolysis, which breaks down the triglycerides. It inhibits the acetone, which decreases the free fatty acid formation, and then which results with more production of glucose, which substrates for metabolism. The regulation of the glucan release, increased blood glucose levels inhibit glucan release. Amino acids stimulate the glucan release, which means high protein and low carbohydrate meal. And then you have stress, and insulin inhibits glucan secretion. Lastly are our references. My name is Antoinette Adams. It has been a pleasure giving you this presentation. I hope that you have gained more insight of how the pancreas works in the body and how diabetes type 1 and 2 are formed from either high glucose levels or extremely low glucose levels. Again, my name is Antoinette Adams and it has been a pleasure teaching you.